Hello, everyone. My name is David Ruck. I'm at the University of Maryland in Baltimore, Maryland, and this is a re recording of my CYTO2026 Parallel Talk, a semi supervised pipeline for a comprehensive and scalable analysis of the immune heterogeneity in human samples. So, spectral flow cytometry is amazing. We have an increasing number of fluorophores. We can acquire millions of cells in a few minutes. This allows us to characterize rare cell subsets and expand our knowledge of human immune system heterogeneity, and it's particularly invaluable for studies with limited biospecimen. However, it does come with increasing analytical complexity. So if you're only interested in a single population of cells, the analysis process remains essentially unchanged. However, if your goal is to characterize all the cell populations that may be present, the breadth and the depth will complicate the analysis. So if we step back and think about this globally, if every marker in your panel split into a positive and a negative population, and you repeated the splitting process for every single marker, and then grouped the cells that shared the same expression of markers, at the end, on the high end, you would have for a 9-color conventional flow cytometry panel, 512 groups at most. By comparison, for 40-color spectral flow cytometry panel, you would have over a trillion of these groupings, and for a 60-color panel, you would have over a quintillion of these groupings. Now, fortunately, biology is not exactly math, so there's actually a lot fewer than this, but it's still a lot more than what you could do with the supervised manual analysis. So at that point, we need to be able to implement unsupervised analysis to be able to do this. Now, there's a lot of variation as far as which approach we do, but there's some areas that we all agree on. We all agree if the unsupervised analysis is resulting in every cell being placed in its own cell cluster, then that is clearly wrong. And vice versa, if all the cells are being put together in a single large group, that is very much not correct either. But then at that point, the different methods for unsupervised analysis go somewhere back and forth between these continuums, and we're just trying to make sure, are they actually grouping things that need to be grouped together correctly, or are they splitting things that should not be split at that stage? And this is particularly important for the context of my PhD work, where I'm working with HIV-exposed uninfected infants. So these infants are born to mothers who are living with HIV, and during the course of their pregnancy, they're exposed to both the maternal virus as well as the viral proteins, but they don't become infected. They're also additionally exposed to antiretrovirals or a combination thereof. And the reason we're interested in HIV-exposed uninfected infants is they're seen in the clinics during the first 6 months of life with substantially higher rates of lower respiratory tract infections as well as diarrheal diseases compared to their unexposed counterparts living in the same communities. So, working with our collaborators out of Malawi, which is in southern Africa, we've been following a cohort of these infants and their mothers, including moms who knew their HIV status before pregnancy, had been on ART throughout, and therefore had pretty close to undetectable viral loads throughout the pregnancy, as well as moms who only found out their HIV status during that first antenatal care visit, at which point half of the pregnancy the infant would have been exposed to high viral loads and increasing amounts of ART throughout the second half of the pregnancy. And then for our study timeline, we collected cord blood as well as 4-month and 9-month peripheral blood draws to be able to monitor how the immune system changed over time in response to the childhood immunizations. And then ultimately on our end, we're processing it with a 32-color spectral flow cytometry panel that includes markers we need to characterize out most of the T cell subsets, as well as also be able to measure cytokines and a few activation-induced markers. And then ultimately, we're processing it very typical for many of these flow cytometry experiments. We are thawing the cells, we are resting them for a little bit before we stimulate with antigens, including PPD and SEB. And then at that point, once everything's gone through the simulation process, we then go ahead and process for spectral flow cytometry and acquire the day of. So, given the uniqueness of this cohort, my personal goal was I wanted to leave no cell population behind. So I wanted to be able to fully profile the entirety of our acquired data set, not just a single cell population of interest. Now, fortunately, we had a trial run to this. We looked at the unconventional T cells using a previous panel, and this taught us a lot of lessons. The main difference for this particular cohort is, at this point, we're trying to shift from where we had been for the previous paper of a 50-50 Flojo to R split to increasingly using more R. R is not the fastest programming language, so we've done some targeted rewrites in Rust to be able to speed up the original R code, and then we are building around the idea of reproducibility from the very start instead of trying to implement it after the fact. And then we've also been trying to have improved bioconductor interoperability to be able to move the bioconductor packages afterwards for statistical analysis. So in terms of our pipeline, we are acquiring samples on a 5-laser Cytek Aurora that has passed both our core as well as the manufacturer QC recommendation criteria. At that point, we are screening our mixing controls for the presence of tandem degradation as well as autofluorescence contamination. We're doing this with our R package Lucianica, which basically, at the level of individual cells, is grouping cells that have the same normalized fluorescence signature based on shared peaks of relatively similar heights. What this allows us to do is we can spot when the antibody tandem starts breaking down based on the variant signatures that are produced, as well as be able to notice when it's a situation where the fluor-fluor antibody combination is not sufficiently bright enough and there's residual autofluorescence being left in the actual single color signature, that we can target those particular fluor-fluor replacements from using cell controls and replace them when you use bead controls. At that point, we're unmixing agnostic. We can unmix in SpectraFlow using these modified outputs that we're getting from Lucianica, but we can also unmix entirely in R, either using OLS, which we're using via the Lucianica package sped up by Rust, or alternatively TrueOLS, which we've been using through the Julia wrapper that I wrote, as well as the autospectral package. At that point, once we have gotten through unmixing, we want to still make sure to screen for fluidic or laser-related issues. So in R, we can use PicoQC or FlowAI, but I also recently learned that PicoQC has been implemented in Rust, which is also a little bit faster. Once that's done, we need to go ahead and clean out the doublets, debris, or the dead cells. We can do this in either R or FlowJo, but then at that point, we're relying heavily on automated gating within R to gate down for the major cell subsets, using either the Open CYTO package or more recently the FlowMagic package, which Sebastiano has been working on. And at that point, anything that's being automatically applied, I want to visualize to make sure that the gate is correct. So we're having all these intermediate outputs get sent to PDFs that allow us to be able to easily screen. And then at that point, since not everybody on our team works in R, it's nice to have a FlowJo version 10 intermediate. So we're exporting things at this stage from R to FlowJo via the CYTO-ML package in its Docker implementation. And then at that point, if there's any gate that's still off, we can just manually adjust it before we go downstream for the rest of the analysis. And then at that point, with all the major cell subsets gated, we go ahead and export out those statistics and proceed for the analysis for them. At that point, we're no longer working so much as iterating through individual experiments, but we're now iterating by the cell subset of interest, like CD4, like CD8, like CD4 negative, CD8 negative T cells. So basically, at that point, from that FlowJo workspace, we can export out the cell populations of interest as their own individual .fcs files, which reduces the total amount of memory being used for the analysis downstream. These get brought back into R using the Flow workspace package. And then at that point, this is where our pipeline differs from others that have been previously mentioned as far as our use of our own R package Coerba. And the basic idea about Coerba is it's basically trying to append to each individual cell a manually annotated gating information. And the way it's basically targeting this particular spot within the spectrum, where it's able to identify out cells that share every single marker with each other. And the way this is basically done is we're relying heavily on Open CYTO behind the scenes. Basically, we're implementing using the min density function, trying to estimate where that split point between the positive and the negative cells is. And if it's still not quite right on the automated gating, we set some gate constraints, we check it again. And if it's still not right, we open a little Shiny app and just do a manual intervention at that point. Once at that point, all the actual gating information is correct, and we can delineate between a positive and a negative cell population. At that point, this gets broken down like just the same kind of grouping by shared marker expressions. And this manually annotated information gets appended on as a new column in the FCS file, with the updated information also stored within the keyword description list. And in turn, we can then pass it on and create a summarized experiment object, which is used by Bioconductor, especially the statistical packages downstream. So this not only permits us to do the statistical analysis, but also allows us to filter for these kind of groupings of cells that may only be present within one category of our metadata. So like return to groupings of cells that are only being seen in HU high infants across many different experiments, or return like individual groupings that are only present in one particular individual, which may be a batch effect. And then at that point, having terminally grouped all these little populations of cells, it's relatively easy to reaggregate back and get back marker expressions on a per individual basis, kind of similar to what we would have done if we had manually gated from the start. And then at that point, we have the option of going into a fully unsupervised pipeline. We can normalize the cells either using cytonorm or cycombine. We can cluster the cells, typically using FlowSum, or we can go for dimensionality visualization, typically using either UMAP or PackMap. But unlike the other kinds of steps as far as that goes, that Core ID column containing the manually annotated gating information is still present. So at that point, it's relatively easy to just gate on a particular island or peninsula of interest within dimensionality visualization, rederive out the marker expression on a per individual basis of those cells that ended up within that island, and then get back the same kind of information we can visualize in a heat map table. So it's not entirely just a qualitative assessment, but a little bit more closer to a quantitative assessment. Now, this approach has been very functional and has been iterative, and there's plenty for everybody to dislike, regardless of if you're in the supervised manual analysis camp or the entirely unsupervised analysis camp. However, there has been some very fun off-label use cases that we've encountered so far. So, because that manually annotated gating information is present on each individual cell, we can use it to check assumptions as far as our unsupervised methods, like is their normalization performing similar for all groups of cells? Is the clustering actually splitting or grouping things that we would have done so manually, et cetera, et cetera. And then there's also an added benefit of this looking at the entire forest approach, wherein we're relatively easily able to identify badly cryopreserved specimens where the cell population was lost entirely. And with that, I would like to first and foremost acknowledge all the mothers and their infants and their communities without who this work would not be possible. I'd like to thank the University of Maryland Greenbaum Comprehensive Cancer Center's Flow Core for the use of their instruments. I would also like to thank for the Bioconductor Project both Hervé and Vince, who were immensely helpful in walking me through how I would implement an S4 system to network with the Coerba package, as well as all the cytometry R package maintainers, past and present, without who this pipeline would not be possible. I'd also like to thank the Molecular Microbiology Immunology graduate program, as well as my thesis committee for all their help throughout this process. And with that, I would like to take any questions. Here's the different QR codes to the different R packages that we have implemented that haven't been mentioned or not as extensively mentioned in the course of the slide deck. The slide deck is also available online. And separately, if you think this is great and you'd like to be able to do it on your own, but you do not know R, since February, we have been doing a free weekly virtual cytometry and R course aimed at those with no to little coding experience. And at this point, we're sequentially working our way through. We're at the end of the cytometry core portion, about to go into the unsupervised analysis. So it's going to be very similar as far as the material being covered by our pipeline, but just in more depth. And with that, I'd like to thank you for your attention.